Björn, welcome to the Beginner's Mind Show. I'm really happy to have you here today on the show to talk about your biologics approach with Eleva, your company. And you have a tremendous experience in life science and biotech as the CEO of Eleva, a company that's, I don't know if it's really quietly, if this is the right word that I should choose, but you move your company forward from a platform company to a pipeline company, are backed by, found it on the internet, 60 million Series A round and focus on a huge problem in the industry that needs attention, but in my opinion, probably is a little bit underrepresented. And I'm happy to have you here to hear more about what you do, what your team is moving forward, and why it matters today.
Right. Okay. Good afternoon, Christian, and thanks for having me. It's a pleasure to join your podcast.
Yeah, absolutely, absolutely. I'm looking forward to the next 60 minutes to torture you a little bit with my investigative
Yeah.
questions.
Right.
Let's start with the first one. What's a question you wish people asked about Eleva but never do?
Oof, that's a good one. So I believe a question that we are asked sometimes is why didn't we hear from this technology earlier? Why has Eleva kept it so quiet for so long? And it's a rightful question, and the answer is rather complex, as always it is in life. But the main question is that it took a very long time to get this technology from the first baby steps when it was spun out from the Freiburg University to where we are now, industrial scale GMP 2000-liter production. And the time in between it looks rather long, but if you compare it to other technologies, even to CHO, then that's the normal time frame you need for that. And the company was flying under the radar for a very long time because the company has the quite luxurious situation that we have as an investor who is very dedicated, who is very loyal to the company. He went through the good times and the bad times, and it was not so necessary for the company to go public. So the company for a very long time had the luxury to work in quiet and optimize the technology in peace.
It's always good to have an investor who is aligned with the company's mission and supports the team. It's good to hear that these kind of investors exist.
They exist, absolutely, yes. And we have this situation. It is the Mr. Dieter Schwarz as person. He likes the company a lot and he supports us. And he invested mainly through one of his investment vehicles. The 11Z is the name of that fund. He invested until now more than 120 million euro into the company over the time.
Wow. Wow.
Yes,
That's
yeah,
impressive. That's
yeah,
impressive. I mean, I was already impressed by your Series A 60 million announcement, but 120,
yeah,
that's really, really massive. And there is a lot of value in your company, I believe. Before we move forward,
sure.
Um, since I heard, sorry for using the word torture before, but since I heard Jensen Huang's NVIDIA CEO speech where he said he tortures people to greatness, I somehow picked it up. And
Yeah.
I also know that executives like you and Jensen Huang always are short on time. And I know from my audience, a lot of investors and executives, that some jump off after 5 minutes. My next question to you is for those type of audience who says, I'm so busy, I need to come back later. What three takeaways will they find in the next 60 minutes?
Three takeaways. Okay, good. The one thing is the first takeaway, if you have a protein that you want to develop to a therapeutic, but you have problems to work with CHO, which happens sometimes. It happened to me when I was young and working with CHO. Don't despair. There are possibilities to come over that. That's the second. The first. The second would be is that one of the promising technologies is ours. So take a look at it. I mean, Of course, I'm pitching this now, but seriously, when I joined the company for a reason 2.5 years ago, and one is that I was really convinced that this is a very, very impressive technology. And the third one for people who have not so much time is a very general advice: take more time for yourself and not only for your work.
That's good to hear. And now let's get right into your technology. I read in your material on the internet that you consider biologics production is broken. Why is that? Why does it need fixing?
Yeah. So if you look back like 20, 25 years ago, CHO technology became the mainstay, the workhorse of the industry, so to speak. And rightfully so, because for the majority of proteins, this technology works quite well. Not optimal in all cases, but it works well enough. In some cases, it works pretty good, like antibodies, for example, usually work fine. But as soon as you get a more demanding protein you want to develop, it can create difficulties with CHO, especially if this is a large molecule or molecule that's complicatedly folded, or that needs certain pH or temperatures that CHO doesn't like to be expressed, and so on. Then you have a problem. And I've seen in my career really pretty and interesting projects die left and right just because the colleagues were not able to bring it to work in CHO, or were not able to upscale CHO, which is also sometimes a problem. It works fine in smaller volumes, but as soon as you go to the large tanks, then you can get in trouble. So this is a problem that many of us have seen over the last decades repeatedly. And it was always a source for frustration on the one hand, but on the other hand, also a source for innovation. And if you look at it, if you go to technical conferences like PEX, for example, you see always scientists that show small step better solutions for CHO cells.
Mhm.
And these are always quite small steps forward. And when I've seen then 2.5 years ago this technology from Eleva, I was hooked immediately because it's very convincing, and I believe that we will not replace CHO, of course not. For that, it is too long in the making, and it's too successful in the most cases. But I believe our technology offers the possibility to salvage projects that might otherwise die.
Mhm. That's good to know. What you said that you immediately got hooked 10 years ago by the technology of Eleva. What drew your attention that you said, that's it.
Yeah,
That's
yeah.
that's moving the industry forward.
Yeah, it was basically two main points of the technology that immediately I found extremely convincing. The one is that the technology follows process-wise
Mhm.
exactly the same steps as the CHO technology is doing. So every step in the process, as well as the equipment, is basically identical. So that was a very clever step from the Eleva team long time before me when they worked on it. They developed it from the beginning, absolutely parallel to what they found with CHO, so
Mhm.
that people who wanted to work with the technology would not have problems to adapt it to their already existing systems, right? So I found that very, very sexy. And the other thing is that they showed me data from a couple of internal projects where they could show me that proteins that did not work in CHO worked very well in this MOS-based
Mhm.
technology. And one of these projects we just now bring into the clinic, first humans should be should be injected within this month still.
Yeah, that's a smart move. So basically, if I understood you right, your team had the approach to simply use existing processes and plug in another technology and not do something completely new that needs a lot of work to be
Yeah.
compatible with existing processes in your customers' firms.
You're completely right with that, Christian. So the point is that it looks like a CHO process only with a different organism.
Mm.
That's it. And because this organism is a plant, it needs light. We still need light in the process. We are working on that as well. But and Sartorius, who is the maker of the tanks that we use or the bag
Mhm.
holders
Mhm.
that we use for large scale production, they were a kind collaboration partner, and they developed or they modified their standard tanks in a way that we just have a light source on the inside. Otherwise, it's exactly the same. We use the same bags, we use the same tank, just with a little light source on the inside. That's it.
Do you understand it right? So, for example, if I would like to produce a protein for developing drugs, then I
Mhm.
can use standard technology available on the market, and with your technology, I have an alternative if the first approach doesn't work.
Correct. Yes. Yes. So we have seen it again and again and again that we can make proteins that do not work at all in CHO or other systems even, or that have in CHO, let's say, a very unsatisfying yield, where you get some protein out of it, but not
Mhm.
enough that it makes sense to upscale it to an industrial scale. And then you can try it in our system. And we have a lot of examples where we could show that it works. We have now also an internal project, which I find very interesting. Our CSO, Andy Schaaf, um, is working on that with his team, where we have a systematic approach showing that a number of CHO non-producible proteins work in our system. It's a long story, and I'll try to make it short. So, a couple of years ago, there was a publication where people tried to express the human secretome, so all the proteins that are in your bloodstream, that they are produced in small scale CHO. Uh, um, and from about 2,700 proteins, 900 failed. Either they were not able to be expressed in CHO at all, or the yield was very, very bad, or the proteins were not produced in the right way. So wrong conformation or something like that. And we could show that we are working on this now. It's still a work in progress, but we can show that we can at least more than half of those that failed in CHO, we can produce without any further steps to optimize the process. So in the first shot, so to speak, we could make around half of that. So this is still unpublished data, and it's still work in the making. But this just shows the potential of the technology, because just imagine out of these 900 might be a couple of proteins that would be interesting for therapeutic purposes, right? And if you can't make them with CHO, then but you can make them with our project, with our production system, then I guess that might be a good thing.
You would make investors very unhappy when you say we have a protein that works, we can put it in a drug in the lab, but we
Mm.
can't scale the production process. This would
Yeah,
be
it's
very
it's
unfortunate.
horrible. Yeah,
Mm.
it's horrible. It's if you imagine, usually that comes relatively late in a project when you are also on the brink of bringing it to the clinic when such projects fail. So imagine how much time, love, and money has gone into such a project, and then you have to discontinue. This is really very bad. So I believe it's in many of those cases it might be interesting to search for an alternative. Yes.
Not totally coming from the business side and to make sure that they fully understand it, you not aiming at replacing what already exists on the market. You found that certain part of proteins cannot be produced in the standard processes that are available currently on the market, but your technology can close that gap. You have an alternative now.
Correct. That's our story. It would be quite, well, hubris to say that we want to replace CHO. Who knows what happens in the next 50 years? But
Mm.
in the moment, we as Eleva, our goal is to offer the alternative for such projects that fail otherwise. Yes.
So if if a biotech company or a pharma company has a protein and they said, look, I mean, the existing standard processes don't work here, either we can't produce the protein or we can't scale it, then you can do a pilot project with your team and say, let's try it. If our technology closes that gap,
Yep.
there's a high chance. And we can show you very simple, can we produce it and can we scale it?
Exactly. So that's it. If you have such a project, before you put it in the drawer, give us a call.
Mm. Yeah,
Yeah,
well, it's
correct.
worth it. And I mean, from my business soul now says, I mean, it's a perfect service business, and you are perfectly situated then to benefit later downstream when something hits the market, that you get some royalties back. It's your technology, you have patented, so the standard contracts always work fine in your favor. But before you mentioned that's not enough. We also have our own projects.
Yes,
My
yes.
question is bluntly, like in school, why?
Yeah, yeah. Well, um, this is this. Let me give you two answers to that. So the
Mm,
first answer is we are not positioning ourselves as a service provider because in that business we don't have the scale for that, and
mm,
also margins are very difficult in this type of business. But what we do is we offer the technology to other CDMOs who are established CDMOs.
mm,
And we are quite picky with those partners that we would choose. But we offer such CDMOs the possibility to get access to our technology by partnership and licensing. And basically, the first of that kind of partnership we published in January with the Spanish/Finnish company 3P Biovian,
mm,
which is a well-known player in the field, a mid-sized CDMO in Europe. And we are currently discussing with others that have interest. So we offer this technology to CDMOs to offer it to their clients. That's the one thing. We are not a service provider, but and there you are right. We have our own projects in the pipeline. Historically, we are that old company already, so we can talk like that. So historically, those projects that we have now in the clinic, they developed from showcase projects.
mm,
Eleva in the time before my time, were then developing the technology, and they had to show to the outside world that the technology works. And they had a couple of such showcase projects, and two of them developed then into projects that now are in the clinic. One is alpha galactosidase, which can be made in other production systems, but with, as we know, relatively bad yields and by that high cost of goods, which is a disadvantage. And we have that. We have that now. We got it through a phase one, and are actively searching for partners who would like to work with this alpha galactosidase. There are two on the markets already, one from Takeda, one from Sanofi. But as I said, as far as we understand, their cost of goods are relatively high. And another thing is, now we dive a little bit into the biology of our moss, that our glycosylation pattern that we add to the proteins that are expressed in our system is different from CHO. And in that case, we learned that it seems to be to the advantage of alpha galactosidase. So with our glycosylation, it seems that we have a better efficacy in patients. It looks that instead of 2 weeks between the treatment intervals, we can probably offer longer time frame for the patient between the treatment, and that might be a very interesting feature of our alpha galactose on top of the better cost of goods that we can offer here. The other project is an even more sexy project, and that was the second reason beside the technology platform itself. This project itself was the second reason why I signed then with Eleva 2.5 years ago. This is Factor H. Factor H is I'm an immunologist by training, and it's a protein that immunologists know. It is a central regulating molecule in the complement cascade of the immune system. I don't want to bore you with the details. When I teach immunology at the University of Heidelberg, then my students always hate that part of the immune system because it's quite complex. So to make it easy is that it's very important to regulate that part of the immune system. And if it's not functioning, this protein, as it is the case with the patients, then the immune system at that part is overactive and makes the patient sick. And there are a couple of indications with that. The most of these indications are rare diseases. But one indication is a very large one, which is AMD blindness in elderly. And all of them are in all of these indications, Factor H is central. And that has been recognized already a long time ago in the mid 1980s, late 1980s. People saw that. And at that time, the industry tried to make Factor H recombinantly to get it back into the patient where it doesn't work, right? To get the patient treated by replacing the non-functional protein with the one that now works, bringing it from the outside into the patient. But they failed. So the industry tried all kind of production systems, CHO, E. coli, Neisseria, whatever. Everything failed. Either it didn't work at all or with extremely low yields. And at that time, Eleva tried to do it as well. They said, wow, when everybody fails, let's try it. And it worked. And it worked very well to our surprise. And we were able to generate a lot of non-clinical proof of concept data in these indications or in the animal models for these indications that show that it works, our protein. And we bring it now into patients, and we have our application was for the clinical trial was successful, and the first injection is scheduled for the next couple of weeks.
Congratulations, congratulations.
Thank you very much. We are very excited about that.
I believe that. You mentioned before that everything failed in that indication on the market.
With this protein, yes.
With this protein. With this protein and in this indication, then of course, whenever it failed. And you said that your team thought it's a good challenge and let's pick it up. My question to you is, why did your team decide? I mean, normally when a lot fails, many people would say, I don't touch that. This is a failed area. Why did your team decide otherwise?
Well, that was in the time of one of my predecessors, and I really admire this decision because it was very courageous to do that. I mean, you can always say, oh, let's try that for fun in the lab, right? But you have, as a company, you can do that in an academic surrounding. But as a company, you have to think about such decisions very carefully. And the decision was very courageous. It was risky, of course. They could have failed as well as all the others. But they did that step. And thank God they did.
Mhm.
And thank you for my predecessor at that time that you did that because it was really it brought the company very much forward. You know, the company's value is now mainly driven by this asset. We can say that. It is really a very, very good asset, very interesting, and the industry has a lot of interest in it. We are talking to interested parties all the time, but the decision, I mean, I guess that the guys who made the decision at the time had a couple of sleepless nights before they really did it and made the decision, but thank God they did. It was a courageous step, and as it is frequently in biotech, you have to make courageous decisions from time to time, and you can fail or you can succeed. And whether you fail or succeed, you see usually only after a while and more investment. And until now, it paid out.
I find this especially fascinating from the business point of view. You said that, totally agree. When you do that in a lab or in a research organization, it's a small scale project and you produce your paper and do a little bit. With a company, you always have to have in mind, on one hand, in preclinics and clinics, it gets really expensive.
Yeah.
So there need to be some interaction with investors or public funds. Someone needs to finance that. And when they see a pile of data and they just feed it into AI, I mean, back then it was not there, but now it's there. The AI would normally say, based on the data, don't touch this. But
Yeah.
this would mean,
Exactly.
but this would mean, and your team proves it, that
Yeah.
innovation dies because new knowledge, new processes evolve out of failed ideas.
Right.
My question to you is to sunshine this a little bit more, this decision. Did you have a
Mhm.
chance to talk with your team through back, who were there back then, what they did to manage the risk so that they say, okay, we know we go into a direction where everybody failed. We just don't throw everything in. How did they approach the risk management process?
That's a very good question. So I believe they followed, of course, the standard, what you do, that it was not one, the main project at the time when it was started. There were a couple
Mhm.
of others around, and it evolved over the time in parallel to a couple of other projects to be the lead project. And that breakthrough came basically from very positive animal data. You have to wait that long until you are convinced that could really be a game changer for the company, this project. And then you put it up in the priority list, of course. Before that, you always have to run a couple of projects in parallel to keep as many arrows in your, how do you say, arrows in your hand
River.
as possible. Yeah, exactly. Thank you. So otherwise, the company at that time was, although being funded by a very loyal investor, the company was always very prudent with its with the funds they
Mm.
received. Have to be, and they did a very good job in that time with that.
I totally agree. And I mean, you always have to serve three people, three parties, the patient, the
Yeah.
healthcare system, and the investors. And if you just neglect one, then there won't be a success. You mentioned before, let's go back in the conversation, that proteins produced with your technology in one indication showed better efficacy than other projects on the market. Did I understand it right?
Yes, that's correct. Yeah,
Why? I always thought, I mean, we are talking still about proteins.
correct.
So
Yes.
I always
So
thought bluntly, the end result is the same. So you have the same protein. Why do you see with your technology better efficacy?
Oh, in that case, yeah, that's a very special case. We have to, we have hints on that, why and we have a working hypothesis with that. And our hypothesis is that it is driven by the glycosylation. So, for those who don't know so well, if you produce a proteins at the end, when the protein is finished, uh, um, you get then in the process also sugars on the surface of the protein, and this process is called glycosylation. And each organism has its own typical individual kind of glycosylation pattern. So CHO has a different glycosylation pattern than other organisms. And each organism type has its own type of glycosylation. In our case, um, um, plants are moss is, although it's a very tiny plant, and we use only filaments growing in suspension, but moss is a modern plant from the evolution point of view, and it has all the apparatus for post-translational modifications, as it's called, as CHO, a mammalian cell line has. So there's no difference in that, only in the details of the glycosylation. So in these many years of technology optimization, Eleva also managed to knock out all the plant-specific glycosylation. So they trained the moss not to make a glycosylation like a plant, to forget to be a plant on that in that respect, and they tweaked it genetically in a way that glycosylation would look like we want to look it. And there are still small, tiny differences to CHO, which is not to the disadvantage, but rather to an advantage. But that would lead now very far and very deep into glycobiology. But for this project that you said, alpha galactosidase, suffice to say that the different sugars that we have on our alpha galactosidase compared to the ones that are on the market leads, that's our working hypothesis, then to the fact that the protein is incorporated into the cell where you want to have it via a different receptor. The receptor is grabbing the sugar molecule on the surface and taking the protein into the cell. And the ones that are on the market have a different sugar than we have, and they are, it's funnily enough, incorporated via different receptors, and that leads to a different way the molecule works in the patient. And in our case, it works to our advantage, it seems.
But this could mean as well that although the proteins look similar, they have slight differences between the different
Correct. Yes.
production
Yes.
processes. But this could also mean that for existing molecules on the market or proteins on the market, it makes sense to look into your technology because the small differences could also make some improvements for the patient.
Yeah, that's of course a possibility, but we are in the moment not catering to that so much. That would open a Pandora's box that we couldn't support in the market at all. But theoretically, you're right, and you see. That just demonstrates that there's a lot of potential in this new technology, and I'm sure that this technology will be still successful in the industry when I'm already old and sitting with gray hair somewhere and just watching the world develop further.
When I look at the longevity space, we are talking about 100 years in the future.
Yeah, well, that is really the thing also with other technologies. Over the time they develop further and further, and new aspects are always then recognized with time.
Amazing company. My next question, I touched it previously before a little bit with the term AI. Everybody talks about AI these days. Do you
Yeah.
use AI for your company? What changes with AI?
Yeah, it's a good question. I mean, I personally, I'm always a little bit cautious with new hypes, right? Fashions come and go, but AI, of course, is a fashion that is here to stay, and that will make our life as scientists easier. And that's how I look at it. AI is a powerful tool that will support scientists, for example, in the but it's a tool, and that's I believe very important to keep in mind that especially discovery cannot in discovery AI can uh, make the handwork much easier and can take off a lot of work from the individual scientist, but the human brain is still necessary at the end, and that's good. In our company, we are working with AI, and we have a couple of colleagues that are really very clever with that. I'm too old for it, for these things, but they are very clever with that. And they are now incorporating AI into our R&D processes where it can be incorporated. And I'm very excited to see that. And I'm always sit there like a little boy at Christmas when they show their results. And that's really a fantastic word and a fantastic new world that is opening up for scientists. I find it extremely fascinating.
You have a long history in drug development and worked with a lot of great companies. You are now in a great company. I've also read in your material that you work together with BioNTech, with Sahin and um, the founding team. When we look at AI, what's your opinion? How will it change the drug development process in the future? I hear every opinion. So from it will replace everything and we will
Mm.
do it fully automated to not important at all. What's your opinion?
Yeah, to BioNTech, I never worked with the company, but I know the founders.
Mm,
So, long time ago, we were working in the same lab and rubbing shoulders
mm.
in the lab. But no, well, who can predict the future? Um, when I was younger, I loved to read science fiction novels, and AI. When I think back in some of the books that I read when I was a teenager, they were already fantasizing how much can be automated, and also how much of thinking can be automated. And I'm now amazed that we are exactly there, where these authors a long time ago what they fantasized about. Now it's becoming reality, and I find it's very good because it is there to support our scientists a lot. And as you said, things that in when I was a PhD student might have taken a week to do, then 10 years later took then only a night by the first robots. Nowadays they take only a few hours to be performed. And the same we will see now with the impact of AI in research, and that this will just make it much faster and more efficient to perform a lot of work. It will never replace the human brain. I'm sure about that. At least I hope it will never replace the human brain. I find this is for me an important fact, never to forget that at the end it's always a human who has to decide whether this makes sense or not. And it's also the human who tells the machine what to do and trains the machine and tells them how to learn and so on. That's the thing. Humans can never be replaced and should never be replaced. I believe that's, for me at least, that's a very uh, deep and profound ethical question we all have to keep in the back of our heads when we work on AI and develop AI further. It's a tool and always should stay a tool.
Yeah, I also think what you say is completely right. And when I look back in the last 30 years with all this technological advancement in the digital space, the companies that tried to replace humans
Yeah,
had a very short lifetime. So this,
yeah,
this really didn't
exactly.
it really didn't work.
Yeah, yeah.
You mentioned you like science fiction. What's your favorite book?
Oh, well, I'm looking now over to my bookshelf, to my bookshelf. When I was a teenager or no, an early student, I found The Wheel of Time, the series,
Mhm.
which now became a movie and or as a movie series. I loved that always. I read it many times and when the movies came out, I was surprised that they kept relatively close to the books, not
Oh,
completely,
really?
but I like it a lot, and still I find it fascinating. I push it on my three sons
Mhm.
now, not only to watch the movies, but also to work through a couple of thousand pages of books. Yes.
So the Wheel of Time is worth reading. I have it in my library, but not touched it yet.
Yeah, you
Yeah,
see.
thanks for the recommendation. You worked with the founders of BioNTech in the lab. Now I get it right. What I find interesting in the industry, and I would like to hear your opinion on that, is basic research has its own culture. And in my opinion, it's driven by patents and publications and academic credentials, which
Yeah.
is quite normal. And the ultimate goal of basic research is to uncover more knowledge for human society. When I look then at preclinical development, the goal changes. We
Yeah.
have to have speed. We need to make decisions. We need to bring something to the patient through the preclinical development into clinical development to the market. Otherwise, development doesn't make sense. In the companies I worked and the teams that I worked with, we always had this interesting cultural tension between basic research.
Yes.
We wanted to do a lot of studies to improve the knowledge base, and the development teams who said, forget it, we have a streamlined plan up to the market, and that's it. My
Yeah.
question to
Yeah.
you is, as a CEO, you work in this space for a few decades. How do you
Yes.
handle this tension?
You hit the nail on the head, Christian. This is really, um, and whether it's a small organization like Eleva now, or a large organization like when I was with Roche or with AbbVie, it's always the same. And you hit it. You are completely right. Um, I believe those organizations are the most successful that um, a keep a certain space of freedom and liberty of thinking in the R&D field, in discovery, so that people can play around and have a certain kind of freedom to work. But as soon as a project comes to the point where it needs development, they have to let go. And then it has, as you said, it has to go into the machinery, as I always call it, where it has to be brought forward, straight according to your development needs and strict timelines and everything. And I believe the those companies who can give this freedom to R&D and have, on the other hand, a very well-oiled machinery, and this is the most crucial point, have the ability, a good process of finding the right time point and a good way to hand over the project from the one universe into the other. These are not only different planets, these are completely different universes, right? And those companies who are really able to do this transformation from the one to the other at the right time point in the right way, they are the successful ones.
Are you an advocate for a handover and not inclusion of basic research and development?
I'm an advocate for handover, but on the other hand, the R&D folks that originated the project will always be there in the background. They will always support the project, and their brain will always be needed to be picked during preclinical, during the clinic even. There are always coming questions back, and you have to go back to these teams. But I believe that it is good for both worlds to also stick to their guns because you need personnel with very different attitude and skill sets for both universes, and both skill sets and attitudes have their right to be there, but And there will be always this overlap that I just mentioned, but I believe it's very important to make sure that for the sake of the project can have its good time in both universes and being treated in the right way in both universes then separately. Yes.
I like your metaphor about the universes. This is really
Yeah.
the best description. It's not only different worlds or continents, it's really completely separated universes.
Yes.
And I have also seen a lot of difficulties when researchers move into development, but stay researchers in their hearts,
Yeah.
and a lot of tension. And as you said, ultimately, we need to move projects forward to decision-making points. And a decision can be we continue or we discontinue, and both is fine and both is right, especially in an industry where the odds are staked against us.
Mhm.
Christoph Lengauer said it. He runs Curie Bio and was at Third Rock
Ah, yeah.
Ventures.
Yeah.
And he said that this ruthless focus on developing the right compounds forward without emotions is what made his company successful. Um, how do you convince people that this neutral decision making is necessary to move their compounds forward when they really have I mean, this is really for some, it's really babies that they
Yeah.
carry
Yeah.
a long
Yeah. Yeah.
time.
Yeah.
How do you manage these emotions?
It's sometimes it can be difficult to let go
Mm.
for the researchers. I mean, I started also myself as a discovery researcher at the German Cancer Research Center a long time ago with biospecific antibodies. So I know exactly how you feel to let go of project. It hurts. Really, so I believe it's very important that you give them that this what I said before, that you always have this feedback, that you keep them in the loop, how it's progressing the project, and that you also use their knowledge about the project, about that molecule, for example, as much as you can. At the beginning, nobody knows the molecule better than they do, right? And you always have to go back to them. And if you make it clear that sorry, you have to let go, but we still appreciate your knowledge of the compound, and your knowledge is still needed, then I believe it's not so difficult at the end. But there's always this pain to let go. No doubt.
I completely agree. The ideal world would really be a clean handover. Basic research is out and development takes over. But as you said, you always need the researchers because there are always research questions coming up also in
Yes.
product development. So
Yes.
you really can't divide these universes in between. You always have to this communication, but problem squared. It's not only basic research and development. You also
Yeah.
need to talk with your board, mostly investors,
Yeah.
and you also need to interact with the pharma industry, regulators. My
Yeah.
question to you is, as a CEO, how do you keep those parties aligned? What's your secret?
Oh, secret. I don't know whether I have a secret, but you're right. As the CEO, especially of such of smaller companies, but also larger companies, you always have to have a couple of balls in the air, and you have to try to keep them flying. Um, at one point, and there are several points probably, but one point I believe that is very important is that you are you are only as good as the team that you have around you. I had when I was as a kid with Roche, I had a mentor, and he said to me, "Always find people for your team that are smarter than you in their field."
Mm.
Always, never have the attitude that you want to be smarter in the field of expertise than your expert in your team, and then listen to these experts in your team. So that's what I try to do. I try to have always experts around me who are world class in their field, and on whom I can completely lean and rely on. And of course, there's always interaction and maybe sometimes different opinions, but the point is that like that, I can delegate a lot of thinking to these people, and I can take care of, as you said, for example, of the board and investors and such things. And those teams are the where you give a certain kind of liberty and responsibility to those experts that are better than you in their field. I believe these are extremely successful teams if you do it like that. So I've found over the years that such teams I love to work with because I always love to learn from other people. And I love also to do it that way because I've seen that it seems to be working. I mean, other people might have other as you said secrets or recipes, but for me that's one way to make it work.
Yeah, I completely agree. Hiring smart people into a company that even outshine the CEO
Yeah.
is the right thing, but it's sometimes, I think, emotionally difficult because at the end, it's the question, what role has the CEO then when everyone is smarter than them?
I believe a CEO has to be humble.
Mm.
Okay, on the one hand, as a CEO, you always are pushed into the light. You always have to represent the company. But a good CEO also doesn't when sounds now maybe a little bit cheesy, but if things go right, the CEO points to his team, and if when as soon as things go bad, then the CEO points at himself. And I believe that's a very important job of the CEO because otherwise, oh no, in this way, he can keep harm away from the management team, especially, and that's very, very important. Because as a CEO alone, you cannot run the company, but you have to run it with a good management team, and you have to do everything to empower the management team to do a good job and to protect the management team from harm. That's, I believe, one of the key jobs of a good CEO. And I mean, yeah, it's always important also to let your management colleagues um, get also positive feedback when it comes in the direction of the company, especially when it's something they did. Then you as a CEO don't say, "Oh, I did a great job here," but you would say, at least you would say we did a great job here, or you would say we did a great job here. And by the way, that was a very good project by person X. If you do it like that, then you do it in the right way, and that shows also to the inside of the company that every activity, every work, every effort is worth it that you do it, and that it's seen and that it's also appreciated also by the outside world. So it's important that as a CEO you show your team also to the outside and make them shine as well.
I like your example. I also think praise and compliments are so important. I mean, at
Oh
the end
yeah,
of the day, it's your team doing the work. And on the other hand, when something goes wrong, it's your decision to select that team. That's the core of the problem. But the thing is that I always find fascinating, praise is good for the team. Praise is also good for investors and for everybody outside, but also the CEO is a human being,
yeah.
and there is nobody tapping them on the shoulder. So my question to you is, what do you do to recharge your energy, your batteries?
Yeah, I'm a family guy on the one hand, so I take a lot of energy from the love that I have within my family. I have three sons and a lovely wife, so that's very good for me. The other thing is I try to be in nature a lot and just to be alone. Uh, Once or twice per year, I give myself the luxury for a couple of days to be completely off the grid. And I disappear somewhere in nature, usually diving somewhere, and being completely off the grid for a few days, and that helps me a lot. Yeah, family and nature. These are my things.
We're coming to the end of our conversation. I have three questions left, if I may ask them.
Sure.
Sure. The first one is diving. You love diving. What fascinates you in diving?
Oh, what fascinates me? Well, on the one hand, it's it's always quiet in the ocean, you know. It's peaceful and quiet, and I love nature. I'm a biologist, you know. That's the one thing. On the other hand, you learn when you. I believe that's something that seamen, fishermen, divers, and everyone learns is that the ocean is huge, powerful, magic, and that you learn a lot of respect for the ocean and for nature by that.
Mhm. The second question is, we are walking at a high speed into the future. How do you position your company as a strategic asset in today's pharma landscape?
Oof, yeah, that's a good one. I believe we have two aspects here, and currently we are preparing the company to serve these two aspects by a planned restructuring. So on the one hand, the platform we talked about it. It might be a game changer for the one or the other project. So don't despair. We can maybe help you. And factor H, I believe, is something that will change the life of many patients to the better.
And when we look at the European scale, Europe has a large and rich history in pharma and biotech.
Yeah,
But the world never sleeps, and there are a lot of upcoming ecosystems. The US is doing well, China is emerging, Southeast Asia. There is a lot going on in Saudi Arabia,
yeah, yeah,
a lot of activity in the United Arab Emirates.
yeah,
What's your vision for the European landscape if you could change it with your
yeah.
company and with your success?
Okay, let's say like this. In Europe has a problem with too much bureaucracy and a lack of venture capital. Everybody knows that. And this is something where, for example, then the Emirates have a very different position on that point. However, if you look at basic research, academic research, Europe is still world class and will be for the future. I'm sure about that. The problem is more on bringing these things into the industry, so to speak. But there's still hope. Just today was this announcement that a BioNTech wants to take over CureVac. I believe that's really a big story for Germany, and this shows that despite all the problems and all the pitfalls of European for biotech in Europe, there's still hope and there's still things moving, and still this story just shows that Europe is not what some people say far behind. It will never come back. So I'm still hopeful. But yeah, bureaucracy and lack of VC is making the life of everyone very difficult in Europe.
So this would then be a nice call to action for our politicians to set regulations in motion with less bureaucracy and as an end result, more venture capital. But Björn, thank you very much for this conversation. I learned from you that still investors also in Europe exist that are aligned with the companies and support biotech companies. BioNTech is a world-class example. They had 2 years ago $20 billion on the bank account. But I
Yeah,
think also Europe needs more than one biotech to move the industry forward,
that's
especially
true.
on the venture
Yeah.
capital side.
Yeah, right. Okay, I believe so too. So, capital, the venture companies, they all have filled bank accounts. They are only now hesitant to dish it out. They have to overcome that. Please, there's so many good innovation out there, desperate for money. Guys, do your job and invest. That's my wish to the venture capital world, and to the state. Always reduce bureaucracy wherever you can.
Björn, thank you very much for your vision. Thank you very much for driving the company and your team forward into a promising direction. I learned a lot from our conversations. I'm excited to see how your company and your team evolves in the next 10 years and how you change the European landscape. And
Right.
thank you again for your time, your vision, and for making space for this conversation.
It has been a great pleasure, Christian. Thanks a lot for having me.
Thank you, and I wish you a nice afternoon.